Every year, human beings manufacture hundreds of millions of tons of plastic. It accumulates so fast and in such volume that it creates its own geography, flowing like an actual river through natural landscapes. A massive portion of this is polyethylene terephthalate, or PET. It's the dense plastic used in water bottles, clothing fibers, and fruit packaging. Global plastic production now tops 410 million metric tons annually, and PET accounts for roughly 12% of all that global waste. While recycling is often proposed as a solution, less than 10% of plastic is actually repurposed. Traditional recycling typically uses intense heat to melt the material, which shears the polymer chains and results in lower performance products that eventually reach a landfill anyway. The reason it just sits there is that plastic is a biological anomaly. We have engineered a synthetic material so dense and tightly tangled that nothing in the natural world possesses the physical tools to dismantle it. We are drowning in this material because we have created a biological vacuum. Earth's natural decomposition processes simply cannot recognize the pollution we are pumping out. But nature does have a mechanism for breaking things down. The chemistry of the natural world is supervised by enzymes, specialized biological machines that act as catalysts to accelerate chemical reactions. In 1897, a German chemist named Eduard Buchner proved that living yeast cells weren't required for fermentation. A chemical substance inside them was doing the work. He discovered the enzyme zymase and later helped map enzymes like amylase. This diagram shows the physical geometry required. The green enzyme possesses a unique surface designed to receive one specific substrate, the red starch chain. When it binds to the active center, it lowers the activation energy, allowing water to quickly snap the chemical bonds. In 2016, scientists found bacteria outside a recycling plant that use a similar enzyme to eat PET plastic. Nature is trying to adapt. But these wild plastic-eating bacteria stop working if the temperature rises above 30 degrees Celsius, and they can take centuries to fully degrade a single bottle. Natural evolution takes millions of years to optimize these specific physical interactions. To solve a 70-year-old plastic crisis, we must figure out how to fast-forward biological evolution. To engineer a better enzyme, we first have to understand exactly how these massive proteins fold themselves into highly complex, three-dimensional shapes. For decades, determining that shape required a laborious physical process. Researchers used intensive experimental techniques like X-ray crystallography just to map a single structure, literally building models by hand to visualize the result. To speed this up, the scientific community launched CASP, a global benchmarking competition that challenged researchers to predict how a sequence of amino acids would physically fold into a protein. After decades of marginal progress, artificial intelligence provided the breakthrough. In 2020, Google DeepMind's AlphaFold model successfully predicted static protein structures with near-experimental accuracy. But predicting a static shape is only half the battle. Enzymes are dynamic machines. To do their job, they have to physically move, twist, and survive in hostile environments. Designing that dynamic action, the biological function of the protein, is the next computational challenge. Researchers at the University of Texas at Austin are using AI as an active engineering tool, moving from simply mapping existing proteins to building entirely new, highly functional enzymes. They fed data from weak, naturally occurring PETases into a machine learning algorithm. The model analyzed the physical structure and predicted 5 specific targeted mutations to optimize how the enzyme handles heat and varied pH levels. The result is FAST-PETase, an entirely new variant engineered specifically to be functional, active, stable, and tolerant. In the lab, this new enzyme operated at ambient temperatures and completely broke down untreated commercial plastic packaging in a matter of days. In some tests, it reduced the plastic to its molecular components in just 24 hours. This wasn't just a replication of nature. AI successfully bypassed biological limits to create a robust catalyst customized for a man-made crisis. This extreme breakdown is a process called depolymerirization. FAST-PETase shears the dense, tangled plastic chains apart, separating them into their core individual molecular building blocks, known as monomers. Once isolated, these molecules undergo repolymerization. They seamlessly reassemble to create pristine plastic without needing new petroleum. This molecular loop allows plastic to retain its original value through multiple cycles. Unlike traditional recycling, this process allows the same molecules to be recovered and reused indefinitely. To make this a reality globally, we have to overcome one final engineering bottleneck: expression. It is incredibly difficult to get living cells or synthetic biological systems to physically mass-produce these newly designed proteins at an industrial scale. Designing a molecule inside a computer server doesn't clean up a landfill. To matter, we have to be able to physically manufacture that protein by the ton in the real world. To bridge the gap between the lab and the factory, the industry is turning to a model that works: high visibility, DARPA-style crowdsourcing designed to force experimental technology to survive real-world conditions. The 2025 Align Foundation PETase tournament is doing exactly this. It's an open competition that forces AI teams to build better variants of this enzyme and then physically test those designs against standardized industrial benchmarks. Competitions like this generate the very first shared head-to-head datasets for enzyme expression and durability, revealing exactly where current models stall and where the designs get rough. Artificial intelligence is no longer restricted to generating digital text and images. It is now actively writing the biological code necessary to physically dismantle our pollution, turning protein design into a foundational pillar of climate technology.